Hi guys, welcome to my YouTube channel. Don't forget to subscribe and share.
Hi, Assalamualaikum warahmatullahi wabarakatuh. So in this video, we're going to discuss about meiosis. So recall back in our previous class, we have discussed about mitosis. And remember, two concepts in mitosis? Hopefully you remember. What is the first concept? Okay, mitosis is a type of nucleus division, or we can call it karyokinesis, which will produce two daughter cells, and each daughter cell may have same chromosome number as parent cell. Okay, so mean mitosis, the concept is two daughter cell, and then have same number of chromosome as parent cell. Okay, but how about meiosis? What is meiosis? Meiosis is also a type of karyokinesis, but the concept is different. Meiosis, you need to remember, it may produce four daughter cells. And then each daughter cell may have, not same, okay? It just have half chromosome number from parent. Okay. If parent, the diploid, has 10 chromosomes, so after meiosis, the daughter cell will have only 5 chromosomes. Okay, so the concept of meiosis, producing 4 daughter cells, and each daughter cell have half chromosome number from parent. To end mean diploid, complete set. So means complete set, one set from maternal, one set from paternal. So this end is referred to haploid, mean one set of chromosome. Okay. So in meiosis, okay, this is the definition. Okay, a type of nuclear division that reside in a cell with half the number of chromosomes as in parent cell. And this meiosis occur during gamete formation. What is gamete? Okay, we have two gamete, male gamete, also known as sperm, and then the female gamete, also known as egg or ovum. Okay, and then this meiosis involves two successive nuclear and cytoplasmic division. First stage is called Meiosis One. Second stage is Meiosis Two. We have the cell, okay, must undergo Meiosis One and Meiosis Two because it's going to produce, just to produce 4 daughter cell. Okay, so this is the concept, okay, involve two successive cell division. This is Meiosis One and also Meiosis Two. For haploid daughter cell is the product. DNA and other cell component duplicate only once, which is during interphase 1. So means the DNA replication, okay, if you remember, in the cell cycle, after the cell cycle complete, okay, the mitotic division and then the cytokinesis, it may undergo interphase, okay. So the interphase is the phase where the cell need to prepare itself to divide. So it has G1 as G2. For normal cell, during S phase, they will duplicate its DNA. But then, after meiosis 1, the daughter cell produced will not duplicate it or will not replicate its DNA. Itu beza dia. Okay? And then each daughter cell has unique combination of gene. So, this is the basic thing lah. Okay? This is referred to the cell, okay, and then this may undergo, this cell undergo during interphase 1 or the specific S phase of interphase 1, then the cell will undergo replication, okay, ataupun kita panggil sebagai chromosome duplication, but the best is DNA replication lah. And during this time, okay, first it must undergo meiosis 1 and then followed by meiosis 2, but in meiosis 2 no S phase. Alright, so meiosis 1, the purpose is to separate homologous chromosome. Okay, so here is homologous chromosome. So we're going to separate, so become single chromosome and single chromosome lah. And then in meiosis 2, we want to separate the sister chromatid. So here is the sister chromatid. So in meiosis 2, the sister chromatid separate. Same as what happened in mitosis. Okay, so the term. Okay. The term I already teach you, okay, in our previous lesson. the term for the chromosome. If you see this chromosome have the same length and then the color is different, so it shows this is homologous chromosome. Okay, homologous chromosome. So homologous chromosome, the chromosome pair have same length, centromere position is the same, and the staining pattern that possess gene for the same character are corresponding loci. Example, same shape and size. So, kalau kamu nampak dua kromosom yang sama panjang dia, centromernya dekat position yang sama, so maksudnya dia adalah homologous chromosome. Dia membawa gene yang sama, ciri yang sama tetapi berbeza sumber je. Contohnya yang biru ni datang daripada mak, yang merah ni datang daripada ayah. So, ini kita panggil paternal. Ini maternal, ini paternal. Okay. Alright, so here, this one and this one same length. So they are homologous chromosome. This one and this one same length. So they are homologous chromosome. But they are non-sister chromatid. Why? Because they do not share same centromere. This one and this one also homologous chromosome. Okay, still about the term. Okay. First thing first, you need to familiar with the term synapsis. So synapsis is a process the replicate homologous chromosome pair up, and this is happen at prophase one. So, for example, in our previous slide, okay, we can see that chromosome 1 and chromosome 2 have same length, mean they are homologous chromosome, but they are apart. Maksudnya dia berjauhan. And then what happened when they are become like this? Okay, let's say this is the blue color one and this is the red color one. So when they are getting close like this, when the structure like this, so it shows the synapsis occur. Means the homologous chromosome pairing up. Mula-mula duduk jauh and then dia dah duduk berdekatan, sebelah menyebelah. So bila dah duduk sebelah menyebelah, itu menunjukkan synapsis telah berlaku. So when the synapsis occurred, okay, and the process is only take place at prophase 1. Hanya pada prophase 1 sahaja. And then this structure after synapsis occur, producing structure called bivalent. So bivalent, a pair of duplicate homologous chromosome formed by synapsis. So also known as tetrad. Bivalent because we consider 2 chromosome here. Tetrad because we consider 1, 2, 3, 4. 4 chromatid. So the same structure, but we can use 2 different terms. Okay, then chiasma. So chiasma, okay, when the homologous chromosome pairing up, so has a part where the non-sister chromatid of homologous chromosome overlap. Non-sister chromatid, dia bertindan. Di antara homologous chromosome ni, non-sister chromatid tu, tempat pertindihan itu, we call as chiasma. Okay, we call as chiasma. Okay, and then crossing over is a process exchange of genetic material between non-sister chromatid of homologous chromosome occur at the chiasma and after synapsis. Okay, mesti dah berlaku synapsis, maksudnya bila dia duduk dekat-dekat tu, dia dah forming chiasma, so dekat chiasma itulah menunjukkan exchange of genetic material berlaku. Itu kita panggil sebagai crossing over. Okay. Alright, so interphase 1 which is occur before meiosis. So this is interphase lah. So we know apa dalam interphase? G1, S, G2. So G1, the cell increase in organelle number, increase its volume, increase the protein synthesis, and then during S phase, the chromosome duplicate, or we can say that DNA replicate. So from the single chromatid become sister chromatid, and then at G1, the cell prepare itself to divide. So first is Meiosis One. Okay, which also have 4 subphases: Prophase One, Metaphase One, Anaphase One, and Telophase One. Wajib ingat. Okay, masih juga 4 namanya sama je macam mitosis, but at the end we must put number 1 here. Okay. Okay, so during interphase, the same thing happen. So, saya tak perlu ceritalah benda ni. Sama sahaja. Exactly benda yang sama. Okay, now we proceed to Prophase One. Okay, in Prophase One actually have five subphases lagi in Prophase One. Ada leptotin, zygotin, pekitin, diplotin, and also diakinesis. Okay, alright. So in Prophase One, okay, in Prophase One, this is the thing happen. First, list the event. Okay, what is the event yang kita memang clear boleh nampak. Okay, sama je macam prophase during mitosis. What we can see here, the nucleolus disappear, nuclear envelope disintegrate, spindle fiber begin to form. But the most important event that must be stated during prophase 1, we must talk about synapses occur. What is synapses? The process of pairing up of homologous chromosome. And then the second event that also must be mentioned during Prophase One is crossing over occur. What is crossing over? Exchange of genetic material between non-sister chromatid of homologous chromosome at chiasmata. Okay, this is the two important events that must be mentioned during Prophase One. Synapses occur, which the process pairing up of homologous chromosome. Okay, and then the crossing over occur at chiasma. Crossing over is process of exchange genetic material between non-sister chromatid of homologous chromosome. So, itu sahaja yang penting. Lain ni memang kita dah nampak. Sama macam mitosis. Okay. And then for metaphase 1. Okay, look at the diagram. Okay, learn from diagram. So, who align at metaphase split now? Is it single chromosome like this, which we call as sister chromatid, or this appearance is homologous chromosome? Yes, this is homologous chromosome. So we must be specific. Homologous chromosome align at metaphase split. Okay, but before that, you must mention the event lah. In metaphase 1, we can see that the microtubule attached to only one side of each chromosome. Microtubule attached to only one side of each chromosome. So here is the homologous chromosome, for example. Okay, and then each chromosome, they have centromere. And actually at the centromere has the structure called kinotoko. And during meiosis 1, okay, the microtubule only attach at one side of kinotoko for each chromosome. So look at here. So the side yang dekat dalam ni tak dipegang oleh microtubule. Microtubule tak attach. So apa yang kita boleh cerita during metaphase 1? The event, microtubule attached to only one side of each chromosome, and then the chromosomal behavior, homologous chromosome align at metaphase split. Or you can say tetrad align at metaphase split. Bivalent pun boleh. But the best is guna sahajalah homologous chromosome align at metaphase split. So in Anaphase 1, look at the diagram. Since the microtubule attached at only one side of the centromere or at the chromosome, so when the microtubule shorten, it will pull the whole chromosome structure to the opposite pole. Bila dia hanya pegang satu bahagian saja dan bila microtubule ini memendek ke arah opposite pole, so dia akan membawa keseluruhan kromosom. So what happened here? Do the centromere split? No. So the only separate yang terpisah di sini hanyalah homologous chromosome. So the event in Anaphase 1, okay, you can say that the spindle fiber pull homologous chromosome. Ataupun kamu boleh kata the microtubule shorten, pull the homologous chromosome at opposite pole. Ataupun pull the sister chromatid at opposite pole. Homologous chromosome separate. Okay, mesti cakap ni, homologous chromosome separate and move to opposite pole. Why? Because the microtubule shorten. And then the microtubule only attach at one side of chromosome, so the entire chromosome will be pulled towards the opposite pole. Okay, and then telophase one. So kita boleh nampak siapa yang dah sampai, chromosome reach at respective pole, and then the same thing occur which the nuclear envelope begin to form, the nucleolus start to appear, and then this produce two daughter cell. So at the end of meiosis one, okay, produce two daughter cell, and then these two daughter cell, the chromosome number now is n equal to 2. Dia dah jadi separuh. Tadi kalau kita tengok at prophase 1, 1, 2, 3, 4. 4 chromosome means the diploid cell have 4 chromosome. But after telophase 1, the chromosome, each of the cell has only 2 chromosome. Means dia dah dalam keadaan haploid. Tapi adakah kita capai misi meiosis? Tak lagi. Sebab misi meiosis kita ada 2 tadi. Untuk dapatkan 4 daughter cell, and each daughter cell has half chromosome number as parent. Tapi selepas telophase 1 ni kita baru dapat 2. Jadi tak cukup. Then that's why continue with meiosis 2. So in meiosis 2, sama juga has 4 subphases, prophase 2, metaphase 2, anaphase 2, and telophase 2. So 2 must be in Roman. Tak boleh tulis 2 macam ni. Tak boleh. Okay. So in interphase 2, when the two daughter cell produced after telophase and after cytokinesis, these two daughter cell may enter interphase. Okay, so yes, G1, the cell getting bigger, increase number of organelle, increase in cytoplasm size, then supposed to be in interphase, has S phase, right? But this time tidak berlaku. Because kalau kita ingat, each daughter cell ni appearance dia dah begini. Kromosom dia dah memang dalam bentuk sister chromatid. So that's why dia tak buat DNA replication sewaktu S phase. And dia terus sahaja kepada G2 supaya dia betul-betul ready, size dah optimum, energy storage dah cukup, and then they may continue with meiosis 2. Okay. So you remember in interphase 2 memang tak berlaku interphase 2 dia tak berlaku DNA replication, tak berlaku. So kita tengok sama ke tidak? Macam pernah nampak kan? Yes, memang sama sahaja. That's why lah saya suruh kamu splitkan. Untuk prophase, actually untuk meiosis 2 is exactly sama sahaja macam mitosis. The explanation sama sahaja macam mitosis. Okay, so kalau kita tengok event of course sama. The nuclear envelope disintegrate, nucleolus disappear, spindle fiber begin to form, the chromosome behavior, the chromosome condense and become visible. Okay, so the chromosome begin to move to opposite pole. Okay, and then after prophase 2 is metaphase 2. See, now the sister chromatid align at metaphase plate. Exactly, sama macam metaphase during mitosis. So the event, the microtubule attached to the centromere of the sister chromatid. And then the sister chromatid align at metaphase plate. After that, in anaphase 2, the event, the microtubule shorten, pull the microtubule shorten, the centromere split, then the sister chromatid separate and move to opposite pole. Nampak? Sister chromatid separate and move to opposite pole. Spindle fiber pull the chromatid to opposite pole. So dia tarik chromatid. Okay? Because what? The centromere split. Centromere ni tadi terbelah. Terbelah. Sebab apa terbelah? Sebab tarik belah kiri, tarik belah kanan. So masing-masing tarik, apa yang jadi? Centromere split. So the sister chromatid separate. And then telophase 2 sama macam mitosis. The chromosome reach at respective pole. Spindle fiber begin to form, nucleolus reappear. Then about the chromosome is uncoiled, lengthens, and decondense. So after telophase 2, we have 4 daughter cell. And each daughter cell has how many chromosome? 2. So kita nampak 1, 2, 1, 2, 1, 2, 1, 2. So means matlamat dah tercapai lah. 4 daughter cell and each daughter cell have half chromosome number from parent. And then clearly kita boleh nampak the appearance of each daughter cell is different. This one biru banyak, ini merah, kita boleh nampak different kan. So that's why lah kita dan adik-beradik kita pun berlainan. And then one of the importance of meiosis is to produce variation. So kita nampak. Kalaulah yang ini tadi dia bawa banyak gene mak. Dan gene maksudnya dia bawa gene recessive. And then it pula bawa yang dominant. So kita akan dapat yang mak punya. And then kalau maksudnya ini dia bawa yang maternal punya, yang mak dia punya. Macam mana saya nak cakap? Okay, katakan sebab ini ayah kita punya kan, ayah kita, so dia ada dia punya from maternal side and from paternal side, side mak dia dan side ayah dia. Katakan T besar daripada mak, T kecil dari ayah. So kita tak tahu which sperm yang bawa T besar, which sperm yang bawa T kecil. And then this egg kita tak tahu yang mana bawa T besar, okay, dan adakah dia membawa T kecil. So itu semua adalah kebarangkalian dia adalah sangat tinggi yang kita kata range dia tu boleh jadi macam-macam. So kita dengan adik kita pun kita dan adik-beradik pun berbeza. So this is the significance lah to produce gamet. So untuk memastikan continuity of the genetic material tu lah, supaya setiap generasi memang ada 46 kromosom sahaja. Reduce number of chromosome by half. And then restore the normal diploid number, increase variation. So this is the difference between mitosis and meiosis. The similarity, DNA replication occur once, okay. Cytokinesis occur at the end of the process. And the difference, the number of division is 1 for mitosis and for meiosis is occur twice. Number of daughter cell in mitosis 2, but in meiosis is 4. And then the number of chromosome in mitosis, the daughter cell has same chromosome number as parent cell, but in meiosis, the daughter cell will only have half chromosome number from parent. So means the reduced chromosome number by half, from diploid to haploid. Okay. In mitosis, no synapses occur, but in meiosis, synapses occur at prophase one. There no formation of chiasmata, but in meiosis, chiasmata is formed, which is also during prophase one. No crossing over in mitosis, but yes, crossing over occur in meiosis. The most specific is at prophase one. Okay, cytokinesis occur one, but in meiosis occur twice. Mitosis occur in somatic cell, but meiosis occur in gonad cell, okay, the germ cell. Then the genetic content in daughter cell is identical with parent, but for meiosis, the genetic content in daughter cell is not identical as parent.